Hi everyone, it is I, C. I am remaking this video because the last one from yesterday had a black box covering the screen for more than half the video. So let's just jump right into it. For the past week, I've spent 24/7 researching nCov 2019. I have made several research videos as I went, referencing in detail over 20 different scientific research journals and studies. This is the video I've spent the most time on as it encompasses the facts from all the other videos. I have made it as short as possible while explaining all you need to know. I've also collected all of the studies, all the links to the studies, and put them on my website, and that will be in the link to the description as well. So first, a lot of people have asked me what my education is on virology. I went to school for a couple of years to be my kids, so I've continued to educate myself as much as possible. It's been several years since I learned about virology specifically, but I went and I watched a course of college lectures before making these videos, so I'd have a good fundamental understanding. From the beginning, I have said I'm here to document evidence so it isn't deleted from the internet. Even today, I do not know the truth. I don't know what clips from China are actually related to the virus. All I have are theories and evidence to make those theories plausible. I hope you will send me evidence that proves my theory wrong. I really do. I continue to search for it myself. I made a Telegram channel that we can talk and share stuff on. The link will be in the comments. Personally, I won't be convinced of my argument until more Western virologists publicly theorize that this is a bioweapon. I just think you deserve to know about this possibility so you can choose whether or not you want to prepare. There may be lots of virologists making the argument that this could be a bioweapon, but the media is covering it up, so we don't really know. Even if this is not a bioweapon, you should prepare ASAP. This should be your wake-up call because a bioweapon attack is probably inevitable. Alright, so the first video that I made was on cytokine storms. I came across this post on Twitter that brought this phenomenon to my attention. I've heard about them before, but I just had forgot about them, so I had documented tens of videos of young, healthy-looking people dying on the streets or dead on the streets, and there were a few of the person actually collapsing. And I wasn't sure if these were legit, and then when I got reminded of cytokine storms, I was like, oh yeah, these are probably cytokine storms. So I'll just show these while we go along. When the immune system is fighting pathogens, cytokines signal immune cells such as T cells, and T cells is important, so remember that, and macrophages to travel to the site of the infection. Too many immune cells are activated in a single place. Cytokine storms have the potential to do significant damage to body tissues and organs. If a cytokine storm occurs in the lungs, which is how most of these are occurring, for example, fluids and immune cells such as macrophages may accumulate and eventually block off the airways, potentially resulting in death. The cytokine storm is a systematic expression of a healthy and vigorous immune system. So just because you're young and healthy does not mean that you are safe from the disease. Now it's true that these videos, right here, could be from anything. China is a big country and people do collapse in public sometimes. What makes me suspect that they are cytokine storms caused by the virus is two things. First, everyone in the videos are wearing masks. I know Chinese people often wore masks in public before the outbreak, but I don't think every single one of them was wearing a mask. Second, no one is doing anything to help the people. If I thought someone was infected with a deadly disease, I would definitely be afraid of touching them as well. And third, these were all posted on virus with people, these were all posted on Twitter from Chinese accounts that all claimed that these were people that had died from the virus. And then I'm going to briefly mention this. So cytokine storms are a common reaction to acute seroconversion syndrome and in HIV. So seroconversion is when your body is creating the antibodies to fight off the HIV infection. And during that time, systematic cytokine storms happen. It's really rare because HIV infections are extremely rare in America. So you haven't heard of cytokine storms, but they do happen. It's just that HIV infections are so rare that that's incredibly rare because it's already it only happens like 5 or 10% or something like that, or less. I read that somewhere, but I don't know if that's true. So don't quote me on that. The thing is that during Spanish flu, there was a lot of cytokine storms, and it was like somebody in the comments yesterday said, oh, cytokine storms happen when you're on your deathbed and the flu's been really bad and your immune system is on its last string. That's not true at all. During the Spanish flu, there was people that would have a cytokine storm and die from it within a few hours of catching the virus. So they were completely asymptomatic and they just died of cytokine storms. That happened during the Spanish flu. So it's completely possible that this is doing that. Okay. So HIV infection of the T cell. So we're just going to talk about HIV really quick because it will help you understand the rest of the video. We're going to really talk about HIV later in the video, towards the end of the video, but I just want you to have a basic understanding so you guys know what T helper cells are and all these different things. So the first step to an HIV life cycle after you have butt sex and get infected, the virus attaches itself to a T helper cell and releases HIV into the cell. Fusion or entry inhibitor drugs stop this from happening. So it is possible that we can fight this infection with, it's called PrEP. It's these drugs that people take when they might be exposed to HIV, and they stop HIV from being able to infect cells. So there is a semi-cure for HIV. It's just you have to do it within the first 72 hours of being exposed, and the earlier the better. Conversion and integration. Once inside the T helper cell, HIV changes its genetic material so it can enter the nucleus of the cell and take control of it. Another drug can prevent this. Replication. The infected T cell then produces more HIV proteins that are used to produce more HIV particles inside the cell. Assembly, budding, and maturation. Ooh. Till those are done. The new HIV particles are then released from the T helper cell into the bloodstream, which infects other cells, and so the process begins again. At this point, you are infected with HIV and there's no going back. Okay, so the T cells are like soldiers who search out and destroy the targeted invaders. T cells can produce a substance called cytokines, such as interleukins, which stimulate the immune response. Another word to remember. So T cell activation is a measured way to assess the health of patients with HIV and less frequently in other disorders. The handbooks for dealing with this, for dealing with nCov in China have actually come out and said that we need to be monitoring T cells. So they are monitoring T cells. They are monitoring lymphocytes specifically. So T cells are also known as T lymphocytes. The T stands for thymus, the organ in which these cells mature, as opposed to B cells, which mature in the bone marrow. And B cells get infected by a lot of other viruses, so they're not as big of a deal. Okay, so you guys need to have a basic understanding of how cells are able to enter, or how viruses are able to enter cells. Basically, there's these are the proteins on the outside of the cell, and they attach to the receptor. And then if the receptor accepts that protein, the virus can enter the cell. So all viruses are extremely limited into which cells they can specifically enter. On the nCov virus, this is called spike protein, and on the HIV, it's called GP120. Okay, so most people have heard about this study. It's called Single Cell RNA Expression Profiling ACE2, the Putative Receptor of Wuhan 2019 nCov. This study is the one that claimed that East Asian lungs have 5 times more of ACE2 receptor cells than other races. So should have been here. Oh well. First, the study mentions that 15% of people, the mortality rate is actually 15%, and that is for people that go to the hospital. But this again was a very small study. So there's evidence, like I was saying later in the video, that I think that the mortality rate could be as high as 30 or 50%. I think that would be after weeks or even months of infection, because it could take a minute for them to kill your T cells. So we'll get to that later, but So ACE2 receptors are the ones that the nCov cell needs to enter the cell. So it needs an ACE2 receptor to enter, or this needs to be an ACE2 receptor for it to accept the spike protein. No association was detected between the ACE2 expressing cell number and the age or smoking status of donors. Of note, the 2 male donors have a higher ACE2 expressing cell ratio than all other 6 female donors, which is 1.6 versus 0.41%. We also noticed that the only Asian donor had a much higher ACE2 expressing cell ratio than white and African American donors, which was 2.5% versus 0.47% of cells. This might explain the observation that the coronavirus pandemic and previous SARS-CoV pandemic were concentrated in the Asian area. Okay, so now, let me see where I am. Many people have said that this study is useless and doesn't prove anything because it was only done on 8 donors. However, I didn't base my video on just the study. A much more extensive study, we can turn this off now. A much more extensive study with hundreds of different human subjects was done by the 1000 Genome Project. These scientists discovered that certain single nucleotide polymorphisms were associated with a higher level of ACE2 receptor cells. These are unique sequences seen in the human genome. So each of these is a sequence. And basically in your genome, 99.9% of your genome is identical to all other humans. And then 0.1% are unique. And most of these unique sequences only have 1 letter difference. So these are all unique sequences. And a certain percentage of all races share these genomes. So different races have more of these unique genomes than other races, and then for most people it's still just the normal sequence and not the polymorphism one. So you can see that these are all, so basically all these sequences are the ones that are associated with a higher level of ACE2 receptors in your lungs. So as you can see, the top one in each of these is East Asians. And then the other ones kind of switch around. And so we don't know exactly how these different things, like this one could make your ACE2 receptors way more prevalent than this one does, or this one does, so then that number is even more important. That's even a higher number, if that makes any sense. But the people that made this also made this. And this shows basically like 92% of the sequences that were associated with ACE2 receptors were found in Japanese people. So you can see how this goes down the list. And that doesn't mean that these have 50% of what these have. It's just the number of sequences. And like I said, we don't know exactly how those sequences influence each ACE2 receptor. Okay, so this is the study that goes against this study. So this study was published, well, not really published. So this was put on preprint. It's not a study yet. It's just like a hypothesis. It claimed that there was no difference in the ACE2 receptors for any racial group at all. So first, this is a preprint, which means it hasn't been peer reviewed. It's kind of like a draft. And I found the methods were a little flawed. First, there's only one writer. He does have a PhD and he works in genomes, according to the University of South Carolina. But the results of this study is a little ridiculous because this is the only study I've seen that contradicts that East Asians have way more ACE2 receptors in their lungs than any other study I found. So he said, no significant disparities in ACE2 gene expression were found between racial groups, Asians versus Caucasian, age groups, or gender groups. That's kind of crazy because, that doesn't make any sense. However, we observed significantly higher ACE2 gene expression in smoker samples compared to non-smoker samples. Did I save this? Okay, well, basically also in his in his study, he mentions that they only did this study on people with lung cancer. So it's not a very good sample, I don't think, as people with lung cancer. So this article right here brings up a point that is a very good argument against this study, and that is, thus 2 coronavirus cases were just confirmed in the UK. But if you read the article, both turn out to be Chinese nationals. Similarly, a recent headline of the Helsinki Times read, "First case of Wuhan coronavirus confirmed in Finland." But it turns out that that it was a Chinese tourist in Lapland, not a Finn. In fact, as far as I can tell, all of the confirmed cases have been of Chinese people. Now, I know that's not true. I'm pretty sure there's at least a few people that were not Chinese or Asian or Japanese that have gotten it. But from what I understand about it, it was very, very mild for them and not a big deal. So, potentially in line with the findings of Chinese researchers, the mortality rate for SARS in Europe was almost nil. Minuscule numbers of people caught it, and only one of them died in France, where there were an atypically high 7 cases. By contrast, in Hong Kong, the mortality rate for SARS was 17%. In Taiwan, it was 10%. In Canada, it was 17%, but it seems pretty obvious that they were all Chinese Canadians. And so another thing to note is, I didn't know this, but apparently SARS, it's not nearly as contagious, clearly, as this new virus is, but it was spread through coughing and sneezing and everything too. So it's not like it was incredibly difficult for this to have spread in other countries. But essentially, it can be said that SARS was an East Asian disease, which, like corona, can be traced back to bats. SARS didn't severely impact the rest of the world, despite people definitely catching it. It disproportionately killed East Asians. See SARS, the immigration dimension. Okay. Many Chinese believe that, the effect was so pronounced that Wikipedia reports many Chinese believe that the SARS virus could be a biological weapon manufactured by the United States. Personally, I still think that's a possibility. Alright, so this evidence right here with the ACE2 receptors, it should give you the most help for the future of the world. As this disease might have been designed to genocide Chinese people while not harming other populations. And personally, I love Chinese and Japanese people. I talk with them on Twitter. They're hardworking, funny, and intelligent. Most of them actually love Trump and Republicans and democracy. They post amazing memes. And I haven't find a single one that isn't based. They all hate the Chinese Communist Party. But a lot of Chinese people are stuck behind a very rigorous firewall. So we don't have direct contact with most Chinese people. Okay. However, our government despises China. For the past 2 years, Trump has done everything he could to encourage businesses to move their factories out of China, either back to America or somewhere else in the world. He has given all corporations and businesses tax breaks so they have extra money to spend on moving. Then he put a huge 25% tariff on Chinese imports, discouraging people to continue buying from China. I also agree with these actions, by the way. I love Chinese people, but that doesn't mean I hate my own economy. This made most executives in America start to seriously consider moving out of China. Most of them have probably made plans to do so and are just waiting for the reason. This could be their reason. Since January 24th, 90% of exports from China have stopped. The virus has already devastated China's economy, as many corporations will be leaving or buying from other places already. The outbreak has been highly beneficial to America, unless it spreads here, of course. So there's a chance that this bioweapon will destroy China but leave the rest of the world alone. In my opinion, this is highly unlikely. So, next, I did a podcast the other night about this study. It was like kind of an experiment that these Asian people did in Beijing. And it's called Highly Infectious SARS-CoV Pseudotyped Virus, which reveals the cell tropism and correlated with receptor expression. So basically, they made a HIV SARS pseudovirus. So we found that the F5 cells were highly susceptible to HIV SARS pseudovirus. and 5H cells expressed a functional receptor required for the pseudovirus or SARS infection. So F5 and HeLa cells, which is what I talked about a lot right here. F5 and HeLa cells, and also Vero E6 and HU7 and 323, but those aren't as important as these other ones. Factor 5 and HeLa. These are all cells that are called immortal cells. Basically, they don't die. They're cells that just live forever, and they're all human-based cells. Well, they're not all. Well, yeah, they're human-based cells that sometimes have modifications from animals, like the polyclonal antibody one is from rabbits. But so basically, these are used for studying viruses all the time. And here, I'll explain this 1 second. So the pseudovirus was able to infect regular F5 cells via the spike protein of ACE2, while they were unable to infect HeLa T cells via the CD4 receptors. So we found that ACE2 were strongly expressed in F5, Vero E6, and HU7. Okay, so ACE2 were high in these cells, and then these were the cells that the pseudovirus could infect. To further confirm the receptor function of ACE2, we pre-incubated the F5 cells with anti-ACE2 polyclonal antibody. We found that the infection could be abrogated, which means rejected. So basically, they turned the F5 cells into antibody cells, but these are not CD4 T cells, these are B cells. And they found that the infection could still be easily rejected. So these results clearly demonstrated that the ACE2 was the functional receptor of SARS. So basically, and B cells are also, if it could infect B cells, it wouldn't be that big of a deal. Is, for example, 90% of adults have Epstein didn't kill himself Barr virus, which is the virus that infects the B cells. So but the antibodies easily fought it off. So, but the F5, Vero E6, and HU7 cells demonstrate that it easily would have infected and not been easily fought off by your immune system. Epithelial cells, which is what the nCov virus is infecting, and those are the cells of the lung, the ACE2 receptor cells in the lung. So it's just an example of basically what those cells usually are in actual humans and not immortal cells. Okay, so basically with this. So this study found that the SARS-HIV pseudovirus could infect regular cells with the ACE2 receptor. While it couldn't infect HeLa T cells, just the fact that it could still infect ACE2 receptors is a major accomplishment. Most of the time when virologists attempt to bioengineer a new virus, it can't successfully infect anything at all. So even though they were only partially successful, they started messing with this 16 years ago. Do you really think it's impossible that they figured out how to engineer this virus to also infect T cells and receptor cells at the same time since then? Okay, so here we are towards the end. We've got the scary virus, or the very scary study. So this one theorizes that nCov is a bioengineered virus that is capable of infecting T cells just like HIV. And it brings my entire theory together, unfortunately. So first off, we're just going to talk about this really quick. Where are you? Uh-oh. Are they not in here? Oh, shoot. All of my slides, like half my slides got deleted yesterday, and I guess I didn't put this one back. But I had one showing the author of this study. But her name is... I didn't put her name down because it was in the slide. Okay. So she is a founder of an organization. She's a doctor, and she's the founder of an organization, and she's been in practice since 2004. And She's a founder of Sunregan, and it's focused on innovative treatments for patients suffering from neurological, neuromuscular, and ophthalmic diseases. Our first product candidates in development target ophthalmic disease. We further evaluate them for use in serious neurological and neuromuscular diseases. So the good news is that they don't specialize in biological genome or viruses, which increases the chances of them being incorrect, which I hope that they are. But they also did, well, we'll get onto that. So, and with that, we'll start reading the study. So, the new coronavirus, which broke out in Wuhan last year, had a severe epidemic within a month. And so far, scientists around the world have a limited knowledge on it. The picture shows a schematic diagram of the new coronavirus published by Centers of Disease Prevention. So, this isn't from their study. I made this graphic, and there is a structural graphic going around of the SARS-CoV, this is nCov, of nCov, and it's a terrible rendition of it. So this is the good one. This is from those lectures I was talking about that I watched. So it's really interesting that you can see the different cells here, and this one does sort of look like it's bioengineered. It does have pretty much all the same exact elements as this. The biggest difference is in these protein spikes. And so you can see that this is what's GP120 and this is what's spike protein in both these. Okay. The sudden outbreak of Wuhan's new coronavirus spread throughout China and shocked the world. This new virus has extremely high transmission speed, high basic production number RO, and a high lethality characteristics, posing an unprecedented huge threat to Americans. Okay, due to limited information currently available from China, there's a wide gap in the scientific community's understanding of the origin of the virus, the duration of human-to-human transition, and the clinical management of infected persons. Scientists have recently published research papers on the virus, confirming that its origin has nothing to do with genetic recombination in nature and transmission from animals to humans. But there are still many difficulties to be further explored. Origin of new coronavirus seems to have nothing to do with recombination. Most papers point out that the similarity between 2019 nCov and bat coronavirus is 88%, that of SARS coronavirus is 79%, and that of MERS is only 50%. Now, I got a comment yesterday on this that said, no, they're 99.9% similar. Well, all of humans' genomes are 99.9% similar. So when they talk about this big of a difference, they're talking about this is the difference in that 0.1%. So in that 0.1%, they're 80% the same. So 0.08% the same, and then 0.079, and then 0.050. So that's how they actually say that. So natural recombination may not be the cause of the virus. The research findings indicate that 2019 nCov, and I'm taking excerpts from the study, so this isn't the entire study. Like I said, all the studies will be in the down below. Is a new type of coronavirus, overturning the author's original assumption that 2019 nCov originated from random natural mutations between different coronaviruses. That is, they did not evolve naturally. High genetic consistency between patients proved that it has only recently spread to humans. nCov is an RNA virus with a high natural mutation rate, a paper published by blah blah blah, and there. So we're not going to read that because the whole mutation thing is really complicated and not important for you to understand their argument. So first, the virus was first reported in late December when most bat species in Wuhan are hibernating. However, SARS also came out in the winter, so. Second, the South China seafood market, which sells a wide variety of non-aquatic animals, including mammals, it was not sold. Bats were not found there. Third, the sequence homology between these bat coronaviruses in 2019 nCov and its close relatives is less than 90%. Therefore, the sequences of these two bat coronaviruses are not the direct ancestor of 2019 nCov. Basically, they're saying that these two ones that we know about could not have been the virus before it mutated. So this also goes through to explain what we already know again, but it always helps when you're new to something. So, spike protein with 4 precise mutations did not affect the affinity with the human receptor. Each virus must have a receptor that binds to human cells, can only live within human cells, and must rely on human cells for replication. Without these functions, do I do something really sad? I never pressed start recording. I just did almost the entire video without pressing start recording. I know. Well, I'm just gonna go start over again and cry. Love you too. Oh shit, I've been streaming this entire time. Oh, well, that's good news. I thought I wasn't recording at all. Oh, well, hello. Well, yay! I went and complained to my husband because I was like, no, I wasn't recording that entire time. Yay, I was recording. Hooray. Okay, where were we? Sorry. Okay, we're trying to get back to this as quickly as possible. Where were we? Okay. The virus enters human cells through specific surface protein channels. The interaction of viral surface proteins with human cells is similar to unlocking with a key. Okay. So, this explains what we already know. So, previous studies have shown that the different coronaviruses bind to several receptors in different ways, such as ACE2 of the SARS-CoV. The presence of large number of ACE2 receptors in human tissues, especially in the epithelium of the lung and small intestine, provides a pathway for SARS-CoV to enter the body. Previous studies have found that there are 5 residues, in the SARS protein are located in the interface of receptor complex and are considered to be critical size for the SARS-CoV cross-species and interpersonal transmission. Therefore, to our surprise, Wuhan CoV-S protein was found to have a significant binding to human ACE2 despite replacing 4 of the 5 important interfacial amino acid residues, which is replacing the Wuhan coronavirus S in the residue positions, those 4 ones. So 4 out of the 5 vital amino acids changed in the protein did not change its structural conformation. Wuhan coronavirus S protein and SARS coronavirus S protein share almost the same 3D structure and RBD domain, so they maintain similar van der Waals and electrostatic in the interference nature. Therefore, Wuhan coronavirus is still able to pass through the binding pathway of S protein and ACE2, posing a major public health risk to human transmission. We already know that 2019 nCov is a virus different from SARS and that the S protein is highly mutant. Therefore, if the gene sequence, protein structure, and function of S protein of 2019 nCov are different from the SARS virus, it will be insufficient. Curious. However, how can this new coronavirus intelligently and precisely select which site mutations while retaining its affinity for the human ACE2 receptor? Why can the virus change only the 4 out of the 5 amino acids of the S protein? And how do you know how to use these special associated proteins to ensure that these actions occur? So this is showing what genetic engineering looks like versus natural mutation. So it's not like this, small targeted mutation. So you can see this is what they look like. Surprising discovery, S protein infected with HIV. On January 27, 2020, President Pradhan of the Indian Institute of Technology and others published a paper, 2019 nCov Spinous Protein Unique Insertion Fragment of HIV-1 GP120 Protein and GAG, Uncanny Similarity of Unique Inserts in the 2019 nCov Spike Protein to HIV-1, GP120, and GAG. The authors found that there are 4 unique inserts in the spike glycoprotein. Importantly, the amino acid residues in all 4 fragments are related to HIV, the GP120 protein of HIV are identical or similar. Interestingly, although the inserts are discontinuous in the primary amino acid sequence, the 3D model of 2019 nCov shows that the coverage together form the receptor binding site. 4 unique inserts found in 2019 nCov, all of which have identity or similarity to amino acid residues in key structural proteins of HIV-1, which are in nature is unlikely to be accidental. Pradhan and other authors added, to our surprise, the sequence inserts was not found in the S protein of SARS coronavirus or other proteins of the Coronavirus family. This is a shocking discovery because the virus is impossible to obtain this unique insert naturally in a short time. Unexpectedly, all inserts were consistent with human immunodeficiency virus, HIV-1. Further analysis revealed that the alignment of HIV to 2019 nCov was derived from the surface glycoprotein GP120, amino sequence, and GAG protein, amino acids. See there, yeah. The GAG protein of HIV binds to host cells, packages viruses, and forms virus-like particles. GP120 recognizes the host by binding to the receptor CD4 cells, plays a key role in this process. And the binding can induce rearrangement of the GP120 structure, creating a high-affinity binding sites for driver co-receptors, such as CXC4R. See, that's so hard to do. CCR5, like letters and numbers mixed. It is well known that CD4 cells are essential for human immunity and are direct targets of HIV. HIV attaches to CD4 cells and then enters and infects them. The virus then turns each infected CD4 cell into factory, making more HIV, until eventually all CD4 cells are destroyed. People infected with HIV lose their immune or defense system, much like a country without military power. The authors once again emphasize that it is unlikely that the S protein of 2019 nCov is inserted with a fragment that is highly similar to HIV's GP120 protein and GAG protein. The accidental mutations in nature are randomly distributed over the entire length of the S protein, and all these insertions occur at the same time as the binding site of the S protein, which is unlikely to happen. So these are all the inserts. Looks pretty spooky. The mimic homothermic spinous protest glycoprotein of 2019 nCov virus inserts from the HIV envelope protein have colored beads shown as the protein's binding site. Illustrates how HIV virus infects CD4 cells. Clinical evidence: patients with cytokine storm blood lymphocytes decreased. So are the findings of experts such as Pradhan right or wrong? If correct, the new coronavirus should be able to invade CD4 T cells and produce corresponding clinical features. The incidence of dyspnea was 55%, and that's difficulty breathing. Median time from onset to dyspnea was 8 days. 63% had lymphopenia. All 41 patients developed symptoms of pneumonia, and chest CT examinations revealed abnormalities. So this is that one study that talks about the 41 patients that was done in December before all of the censorship really started. And then this study was done 3 or 4 weeks after they were admitted. So if they did die a month or 2 after infection, then that's not included in this. So complications include acute respiratory distress syndrome, anemia, acute heart injury, and human-to-human infection. 32% of patients were admitted to the intensive care unit, and 6 patients, 15%, died. Compared with non-ICU patients, plasma levels were higher in patients with nCov infection causing severe respiratory illness, similar to the severe acute respiratory syndrome in coronavirus, and is associated with ICU admission rates and high mortality rates. Although low white blood cell counts are common in viral infections, it is surprising that 63 is the percentage of patients, 85% of ICU patients show a decrease in lymphocyte counts. According to a 2004 study on SARS, the average lymphocyte count in patients with SARS is usually normal. 2 clinical guidelines for diagnosis and treatment of Wuhan nCov, one of which is, quick guide for diagnosis and treatment of new coronavirus pneumonia. Instructions for handling 2019 new coronavirus. The first guide clearly states gradual decrease in lymphocytes, and the second guide emphasizes the importance of monitoring the absolute value of lymphocytes. Therefore, for a certain percentage of patients, observing the reduction of lymphocytes has considerable clinical significance. CD4 positive T lymphocytes make up the majority of all lymphocytes. Although this is not a routine test for patients with coronavirus infection, monitoring CD4 cell counts might be helpful for the 2019 nCov patients. Another clinical feature of patients infected with 2019 nCov is higher levels of serum, cytokines, and chemokines, the latter being defined in Professor Hong's paper as cytokine storm. This clinical feature is consistent with observation of Predin et al. That the 2019 nCov S protein induces structural rearrangement in GP120, creating high affinity binding sites for chemokine co-receptors. So basically a higher level of cytokine storms. It is well known that activating T cell surface receptors can cause a cytokine storm, which has the potential to cause significant damage to organs and body tissues. For example, if a cytokine storm occurs in the lungs, immune cells such as macrophages and body fluids may trigger tissue damage, resulting in acute respiratory distress and possible death. Although the CDC website states that there is currently no specific antiviral treatment for nCov infected persons, there have been several cases of Wuhan patients benefiting from lopinavir and others treatment with anti-HIV drugs. Therefore, more such detailed clinical experience should be shared. So these are all the problems. These are all the things that they went over in this study. No close genetic relationship to other coronavirus. Similar to SARS virus with ACE2 binding, progressive lymphocytopenia, and cytokine storm, high RO fast human transmission with high fatality, empirical treatment with anti-HIV drugs, precise insertions in the S protein without changing the affinity to ACE2 and similar HIV GP120. Okay, so I guess we'll talk about this for a minute. So like I said, this study didn't come from the most reputable source. They did consult, because they're brain doctors, they did consult a couple of virologists and biologists while doing the study, so it's not like they are completely out of their depths. Mainly, it better explains some of the evidence in the genome that this is airborne HIV, and I won't believe it until I see more virologists discussing the argument. So, still though, you don't quarantine 300 million people because of a bad flu, nor do you will people into their apartments, shut down all your factories, or kidnap screaming people off the street, while sometimes killing them in the process. There's still a very good chance that this virus is not a bioweapon or an airborne HIV, but the reaction from the Chinese government is still pretty scary. So, I'm going to just talk about these really quick. These are all from Chinese Twitter accounts. The interview with Steve Bannon at his war room, Mike's pointed out that there are a total of 49 crematoriums in Hubei province, which work 24 hours a day for the past 17 days consecutively. The real number for confirmed cases is at least 1.5 million. The burned bodies are over 50,000. If it is made by white people, then the investigation is likely to be over, and the Americans themselves cannot be blamed. Of course, the yellow race does not have the IQ to play synthesis. I said before that the Wuhan coronavirus has a high affinity with the human body. It not only infects the lungs, but also easily invades other organs, causing infections in the patient's myocardium and other organs. That's heart. Because myocardial inflammation is not obvious and myocardial pain is lacking, sudden death of myocarditis and renal failure in patients without symptoms is often seen on the streets and in hospitals. At that time, many people questioned my claim that there was that there is no scientific basis. Now mainline medical experts have proven this. That's why you need to administer medicine purely to kill people, take your temperature door to door, and get you quarantined if you have a cold and fever. The quarantine is like a concentration camp, so you can never cross the infection, and so you will definitely be infected and go straight to the crematorium. The Chinese have not woken up yet. Sit and wait. So we'll go back to this. So you should be deeply concerned. You should be getting prepared before hysteria sets in. You think that there's unlimited food in America, right? I went to Costco and it made me feel a little silly because of the crazy amount of food there. But there are only a few foods that are super cheap to buy, have a lot of calories, and have a long shelf life. Beans, rice, pasta, bread, flours, etc. If a pandemic occurs in America, these items will be sold out first. Like all food, their prices will most likely skyrocket. Chances are no one will starve in America, but you might go hungry a lot and you might have to spend a lot of money on food. Also, you will need to continue to go into public all the time, as you will most likely only be allowed to purchase a ration amount at a time. Because this virus has a long incubation period, a big breakout of this disease could occur in America any day now. I think there's a good chance it will be contained to China, especially if it was engineered to kill the Chinese. Still, it could happen. People will probably get hysterical fast if it does. So you need to get prepared as quickly as possible. And I'm going to be doing a video about all the items you should buy at Costco, eBay, and a couple other sources. Sorry. I will show you how much it will cost to purchase enough food for 2 to 4 people to last around a month, 6 months, or a year. So keep an eye out for that. I will continue to document what is happening in China and attempting to contact residents there to find out what's really happening. So for the past few days, videos have just completely slowed down a lot from China, and people are speculating that they have somehow cut. So they were smuggling these videos out through WeChat, and the government has somehow cut this off. So people in Wuhan are just no longer able to get videos out at all anymore. So I don't know if that's true yet, but it might be. So we might not be getting a lot more videos out of China right now until it starts occurring in other places or the government falls and the firewall's lifted. So please share this video with everyone you care about. Convince them that they need to get prepared. I'm trying to convince my husband's large family so I don't have to share the small amount of food I'm able to purchase right with them if shortages start occurring. Remember, if they don't prepare, it will most likely affect you, so please share this video. Again, I made a Telegram channel where we can talk and share stuff on. The link will be in the comments. And I also put all these on my website, which is toomuchblood.com. And I haven't really done anything with that website in a long time, but it exists. And you can go there to get all the different links to all the different studies. I also have to put the PDFs that I have for studies that aren't on the internet, or not easy to find. And I have those, and all the videos are lined up there, and everything is there, so you can just share that webpage with your family, and then they'll have everything out in front of them. So it's super easy. So thank you for watching, and talk to you later. Bye-bye.